welcome, and I think we're going to go ahead and get started. For those of you who might not know me, I'm Ruth Karen. I'm a professor in the Department of International Health at our school, and I have been actually putting together vaccine days now for about 14 years. Well, we've had 14 vaccine days, most of them annual. Before we actually talk about that and I talk about our topic of the day, I just wanted to not forget to thank an awful lot of people, because I think of Vaccine Day and then people actually make it happen. So first of all, our panelists, who will introduce themselves in a little while, our poster presenters, of course, our two guest speakers, Dr. Paz Bailey and Dr. Durbin, our three poster judges, Andy, David, and Kauser, Brendan and Anne, always for expert communications help, Liz Meyer, who leads a team from our center who makes all those posters possible, and of course, all of the teams here that support. So as I mentioned, this is our 14th Vaccine Day. We've been really fortunate to have absolutely preeminent people come to speak to us, and I think just by taking a look at this list, you'll have a sense of that as well. And our two speakers for today's Vaccine Day are no exception. They're Dr. Gabriela Paz Bailey and Dr. Anna Durbin, and they're going to be talking about dengue, new epidemics, and new vaccines. And you'll hear a little bit more about them in a few minutes. But I just have to tell you, I thought about talking about dengue, sometime last fall, and I thought it was a good idea last fall. And unfortunately, now I think it's a great idea because dengue has exploded. It's in the news all the time. This is the PBS NewsHour. I believe Dr. Durbin was quoted in this NewsHour. This is a slide that I think Dr. Paz Bailey is going to show. And what it shows is that in the Americas, it's only April, and we've already had, first of all, 2023 we thought was, it was the most cases we'd ever seen in the Americas. Now by April 2024, we've already exceeded that number in the Americas. But the other problem is, places like the Americas and places like Vietnam or Thailand are used to dealing with dengue. If you're used to dealing with dengue, the mortality is not necessarily so high because you know how to give care. But the other big problem, in addition to huge numbers in countries that have always had dengue, is that now we're detecting dengue in countries that have never experienced dengue. And not only in countries that have never experienced dengue, particularly on the African continent, but in incredibly vulnerable populations, refugee populations, populations of children who are co-infected with malaria and dengue. So this is a huge problem. And you can see this is an illustration of Bangladesh dealing with a record dengue outbreak. So with that, I'd really like to introduce our first speaker, who's Dr. Gabriela Paz Bailey. She is chief of the dengue branch at the US CDC. She brings a wealth of experience with dengue and with a number of other infectious diseases, including HIV and many others, to her experience. She is in the CDC, as I mentioned. She's based in Puerto Rico, which is a hotbed of dengue, perhaps, unfortunately at this point, but I think fortunate that we have based the dengue branch in an endemic area. So without further ado, I'd like to introduce Dr. Paz Bailey.
No problem.
So that's not it.
Okay. Just turn that down. You're awesome.
Okay. Well, thank you so much, Ruth, for the invitation. It's really so great to be here. Hopkins has a big place in my heart, especially because when I come, I get to see my good friend, Amita Gupta. And we had a chance to have breakfast this morning. That was really a big gift. And as Ruth said, I'm based in San Juan, Puerto Rico. Usually I present all these pictures of the beach. I had more limited time, so I'm not going to be showing that to you today, but you can imagine a Caribbean beach with the turquoise water. That's where I work. So, yes. So if you want to know more about working and living in paradise, you can talk to me later, because Dengue Branch always has good opportunities for internships and for other collaborations. So I will talk today about dengue in general, the epi of dengue globally, and also what that means for introductions to the United States, and then what we can do to be better prepared for dengue increases. So why should you care about dengue? I'm sure you have seen all the news articles recently, and these are just a fraction of the headlines that pop up when you search for the term dengue. And because dengue outbreaks result in large case counts within a very short time period, these news stories are often accompanied by some pretty dire photos. And these are two that we took in Honduras during a dengue outbreak there that show overwhelmed hospitals and patients in makeshift wards waiting to be seen. And these images can be distressing for us, knowing that our global colleagues are struggling, but also for us in Puerto Rico, because an outbreak was declared on March 25th, and we're in the low season now. So while you listen to this talk, I would like you to think about what is needed to be better prepared for the increases globally, and also for what is going to happen in the US and its territories. So dengue is caused by one of 4 distinct but closely related viruses. Infection with one virus gives you lifelong immunity to that specific serotype, but only short-term cross-protective immunity to other types. And it's primarily transmitted by Aedes species mosquitoes, but there are other routes of transmission that are less frequent and are listed in the slide. And dengue viruses are among the few RNA viruses with global distributions. RNA viruses have high mutation rates due to defective proofreading during replication. And though dengue viruses don't mutate as fast as influenza viruses or SARS-CoV-2, they have relatively high rates of mutation compared to DNA viruses. And the 4 dengue serotypes form a tight group. They have about 65% amino acidic identity among them. And the figure on the right shows a simplified maximum likelihood phylogenetic tree. The proximity and the length of the branches indicates the evolutionary distance between the viruses. And you can see how the Aedes viruses clustered together and the Culex viruses clustered together. And this is a more detailed phylogenetic tree that shows the 4 serotypes. And Bayesian analysis has inferred separate origins for each dengue serotype from common ancestors in different continents. And the virus has dispersed globally through Aedes mosquitoes since the last century, colonizing different regions. And this has brought diversification and also speciation and the formation of these genotypes. There are between 2 to 4 of these genotypes within each serotype. So dengue is the most common arboviral disease globally, and incidence is likely to increase as the climate warms. And this is both through increased transmission in areas that are endemic for dengue, but also through expansion of the Aedes species mosquitoes to new areas. And it is estimated that 6 billion people, or 60% of the global population, will be at risk for dengue by 2080. In addition to rising temperatures, other factors contributing to the increasing global burden are the rapid increases in human travel and also rapid and unplanned urbanization. And this is data from the World Bank, and you can see these increasing trends in blue for travel, and then the line in red for the urban population. So let's move now to talk about risk factors for severe disease and review dengue pathogenesis. So Most dengue infections are asymptomatic or mild, but between 1 to 5% of patients develop severe disease, and dengue can be fatal if it's not treated appropriately. Factors like age or comorbidities can predispose to severe disease. And the sequence of dengue infections is also an important risk factor, with the second infection with the dengue virus being the riskiest for severe disease, which I'm going to explain in more detail in the next couple of slides. So this is a figure illustrating the differences in risk for severe disease based on the sequence of exposure to dengue viruses. The blue arrows indicate the sequence of exposure, and the color of the people are the level of risk for severe disease. So green is low risk, yellow is medium, and red is high risk. And in this example, the person in the image is exposed to different wild-type dengue viruses 4 times in their lifetime, as shown with the 4 blue arrows. And in endemic areas, a child will be exposed several times to dengue virus before adulthood. So the first exposure is usually medium risk for severe disease, represented by the yellow person, but then the second exposure is the highest risk for severe disease. And after the second, the third, and fourth are low risk for severe disease. And the reason for these differences in risk is what we know as antibody-dependent enhancement, or ADE. And in ADE, lower heterotypic antibody titers are ineffective at neutralizing the virus, but they do bind with the virus and then facilitate entry into monocyte cells. And this results in higher viral load, and this leads to increased NS1 production, increased vascular permeability, and this leads to plasma leakage and can result in severe disease. And there have been cohort studies in Nicaragua and in Thailand that have sort of produced important data to understand this. And this is showing the probability of being hospitalized on the y-axis, the titers on the x-axis, and show that sort of peak in increased risk at certain titers. That is, this window for the study was considered between 80 and 120 in the linear titer. And you can see a similar relationship between titer and increased risk for dengue hemorrhagic fever on the right. So turning our attention to the global situation, we always message out that dengue is the most common arboviral infection in the world, but it's really getting worse in recent years. And these are the preliminary numbers from WHO for 2023. There have been more than 6 million cases reported worldwide, and we know that this is just the tip of the iceberg, since many are asymptomatic and many are not reported. 92 countries or territories reported cases, and these countries are in all 6 WHO regions, and 23 countries reported outbreaks. This map shows the countries where locally acquired dengue cases have been reported in the past year. And first, you can see that countries in all regions have reported locally acquired cases, including Europe, which has had increased number of locally acquired cases in countries like Italy, France, and Spain. But you might notice that Central and South America are almost all the darker brown color. That indicates the highest incidence. And these are, looking at the Americas region specifically, we have seen a sharp increase in dengue cases and incidence over the past 40 years. This figure shows the number of dengue cases reported to PAHO by year since 1980. So as you can see on the right, 2023 had the highest number since the early 80s reported, but I added yesterday the numbers for 2024. And as Ruth was saying, with the 13 weeks that have cases reported in 2024, there have already 4.6 million cases reported. And for the entire 2023, there were 4.5 million cases reported. So already the number has been surpassed. So what is the effect of the increased global dengue activity on its epidemiology in the United States? Well, we should first acknowledge that If we're talking about all US jurisdictions, including the territories and freely associated states, we have 6 with endemic dengue transmission. And these are American Samoa, the Federated States of Micronesia, the Marshall Islands, and Palau in the Pacific, and then Puerto Rico and the US Virgin Islands in the Caribbean. And this figure shows the dengue case data for Puerto Rico, American Samoa, and USVI from 2010 to 2020, with the solid lines showing case counts and the dotted lines showing rates by population. So in Puerto Rico, shown in pink, we had large outbreaks in 2010, 2012, and 2013, but then really low levels of transmission after that. The Zika epidemic played a big role because it provided some cross protection for a while, and then COVID, the stay-at-home orders with kids not going to school also supported longer, low levels of transmission. But now things are changing. American Samoa had also an outbreak in 2013. That was USVI in green. And in blue, American Samoa, you can see in the case counts in the blue line that the number of cases were not great, but the population is small, so their incidence was very high. So this is our epidemic thresholds in Puerto Rico. And I just wanted to show you that we're currently in the low season. So usually you don't see a lot of dengue now. It's supposed to be the colder months. It doesn't feel very cold when you live there, but usually we don't have that many dengue cases reported now. And for about 10 weeks now, the counts have been above the epidemic threshold, and this is why the health department has declared an emergency due to the dengue outbreak. So looking only at US states where dengue is not considered endemic, we can appreciate that the majority of the cases, over 94%, are associated with travel to endemic areas. And this slide shows the number of travel-associated dengue cases reported by year to Arbonet, with more than 10,000 cases reported from 2010 to present, and more or less 800 cases on average annually. But I want to highlight that while 2019 was previously the record year with the highest number of dengue cases, 2023 surpassed that record with over 1,500 of cases reported. So in the next couple of slides, I want to reflect on the link between increased travel-associated cases and increases in local transmission. So let's start by asking, where do we find competent vectors in the US? And these maps show the probability of vector presence in each county in the continental United States, with darker red indicating higher likelihood of vector presence. Aedes aegypti is shown on the left, primarily in the southern states, and Aedes albopictus is on the right, covering large portions of the south, the east, and the west coasts. So locally acquired dengue cases have historically been reported from Florida, Hawaii, and Texas, with transmission occurring as sporadic, single cases, or very small outbreaks. However, in the past 2 years have been quite concerning because we had 2 new states showing evidence of local transmission. In 2022, Arizona reported 2 locally acquired cases, and in 2023, California also reported 2 locally acquired cases. So now, naturally, we're all asking about what we should expect if we see transmission popping up in new areas. So to answer this question, I would like to look at data from Florida, which I think is a telling example of the relationship between travel-associated cases and local transmission. So these are the case counts for travel-associated and locally acquired cases in Florida. And on the right, you see a small outbreak of 72 cases that they had in the Keys. And then if you focus on the right, you see the increases in travel-associated cases after the COVID travel restrictions eased up, and how this can result in locally acquired cases, especially in 2023, where they had really high numbers of locally acquired cases. So I think if you have an area with the right weather, with the with low population immunity and with the vector presence, it will be more common for them to see locally acquired dengue. And it's very important that clinicians are aware and consider dengue in their differential diagnosis and test appropriately for it. And when looking at data among travelers in Florida, overall, 5 countries made up 75% of their travel-associated cases: Cuba, Puerto Rico, Dominican Republic, Haiti, and Jamaica. So how can we prepare for increasing dengue cases now that we have reviewed how much dengue is growing globally, how we're seeing more transmission in the US? Let's think about what can be done. And there are important emerging tools. There are new vector control strategies that I will not be able to tell you about, but I could talk for hours about them, like Wolbachia-based strategies, Wolbachia replacement. But they're not approved for use in the United States. And I'm not going to talk about vaccines because Anna will cover this next. But I just want to highlight that no dengue vaccines are currently recommended among US travelers. So I have 4 suggestions that I think are important to prepare for dengue. The first 3 are: encourage bite prevention among your patients and your communities, especially in travelers. Remind clinicians to consider dengue in the differential diagnosis early on in their workup of patients with compatible clinical signs and symptoms, and also educate them on how to test appropriately for dengue. So in terms of my first suggestion, please tell your patients and your friends that are traveling that there are messages in the CDC, in the CDC web pages about dengue transmission in destination countries. So there's a lot of good information that travelers can get from the CDC website. We have also developed plain language public-facing recommendations that I have provided in the blue text that talk about the use of EPA-approved repellents wearing clothes that prevent mosquito bites, and if possible, staying in areas with air conditioning. And the link to access those materials is at the bottom of the page. And then we need to remind clinicians to consider dengue in their differential diagnosis early in their work group. I feel that this one is very important for US clinicians because there are special considerations for dengue that are important to recognize it early and start treatment early. So these are some materials that CDC has developed, this pocket card to guide medical treatment. There are no specific antivirals available for dengue, unfortunately. So standard of care is systematic fluid management, and they're sort of evidence-based WHO guidelines on how to treat dengue patients. Mortality can be very high if dengue is left untreated, but can be reduced to less than 0.05% with appropriate management. And then testing appropriately for dengue is very important. Now that we have been talking to clinical labs in the US, we're finding that IgG testing is what is used for acute diagnosis, and IgG determines past exposure to dengue, but is not used for acute dengue diagnosis. So the tests that confirm dengue infection are NS1 antigen testing and PCR testing, and it should be accompanied by IgM testing. If only IgM is positive, it provides a presumptive diagnosis because there is some cross-reaction with other flaviviruses, but the recommendation is to do both molecular or antigen testing and IgM testing. So in summary, dengue cases globally are increasing. Dengue cases in the US are also increasing, and we have areas of the US with competent vectors and with frequent introductions due to travel-associated cases that may lead to local transmission. And there are several steps that can be taken to be better prepared for the increases in dengue mosquito surveillance and control, encourage patients to prevent mosquito bites, and remind clinicians to consider dengue early in their workup, and then test appropriately for dengue. And with this, I want to thank my colleagues from the dengue branch. I did not include my email, but if there are any follow-up questions, Ruth knows how to contact me. And I don't know if we have time for some questions now.
We do.
Okay.
We do. Thank you so much. I might lead off. I'll lead off with one. And because we're recording, I might ask you to repeat the question because I don't think there are mics. Okay. But I'll just shout for the moment, which is I wanted to ask a question about testing and availability in the US and globally, and particularly in areas where outbreaks are occurring. What is current availability and what's being done to increase availability? Because I'm sure it's not where we would want it to be.
Yes. So the question is about availability of dengue tests. And it's an important question because I don't think we are where we should be. In the United States, public health labs have access to PCR tests developed by CDC. There are 2 that are FDA approved. One test for Zika, chikungunya, and dengue is the trioplex test. And the other one is the multiplex test that tests for the different dengue serotypes. And those are widely available in public health labs because these tests are not commercially available. So CDC provides the kits and sort of quality control with the public health labs network. In clinical labs, as I was mentioning in my talk, there's a lot of incorrect testing going on. There are 2 FDA-approved tests, an antigen test for NS1 and an IgM test by a company called InBios, but those are the only FDA-approved tests, and we don't have a good sense how much they're used. We're currently doing a survey to understand that better. One area that is problematic is that there are no rapid tests available. And I think that with COVID, we have all come to understand how important it would be to have that tool. But there are several companies that produce them. They're available for purchase internationally. Performance varies, but there's some good evaluations out there to be able to identify the good tests. But in the US, none of them are FDA approved. And globally, not all countries had PCR testing capabilities. A lot of capacity building has been done with COVID. So I think more and more countries do have the ability to detect dengue and confirm which serotype is circulating, but that is not used to aid in clinical management. That just takes a long time for those results. And in many occasions, those results do not go back to the clinician. So the guidance is always to treat, if dengue is suspected, to start treatment without waiting for diagnostic tests. And I just want to mention one last thing about the Americas that have a network of labs for arbovirus diagnosis. And with PAHO and CDC support, the capacity has been built for molecular and serological tests to be available. So I think the Americas is really ahead compared to other regions. Yes?
I wanted to ask a question about the risk to healthcare workers. I wanted to ask a question about the risk to healthcare workers. I don't know. In Bangladesh, when we had a major outbreak a few years ago, there were many of the healthcare workers, the doctors and nurses, who developed dengue and died.
Yeah.
And there didn't seem to be an appreciation about protecting the healthcare workers with, even putting bed nets around the beds or even closing the screens. And in fact, we had higher rates in the neighborhood around the hospitals that seemed as though the hospital was actually, accentuating the outbreak. So any comments about healthcare worker protection?
I agree. So the question is about dengue risk to healthcare workers. And I 100% agree that it's sort of an invisible problem that, we don't talk about it enough. there are ways to reduce the risk of dengue, like the use of screens, like the use of bed nets for people that are sick, but they're not broadly used and not in every single hospital. So for the Cuba outbreak that they had recently, 2022, there were several reports of deaths among doctors. And I think that it's an area where we should do more, and we should probably have more targeted messaging around it. So thank you for that comment.
So I have a question. Is this on? Okay. I couldn't tell. Sorry. So my question is, so I know that all 4 serotypes circulate globally, but my question was specifically about the outbreaks that are happening in the United States. If you can speak to if it's predominated by any particular serotype, or if it's been a mixture, and then also if there's a predominance of serotypes currently in Puerto Rico.
Yes, thank you. So in the case of Florida, it's sort of driven by the country that is introducing more dengue through travelers. So the 2022 cluster of locally acquired cases were mainly dengue 3. And in 2023, it has been a mixture of dengue 2 and dengue 3. But in the Americas, dengue 3 is predominating. So I think that is going to sort of guide the introductions to the US. In Puerto Rico, historically, we had dengue 1 transmission. For all the previous big outbreaks in 2010, 2012, and 2013, dengue 1 predominated. But we started seeing a switch in the predominant serotypes at the beginning of the year. And now dengue 2 and dengue 3 are predominating. And the concern is that previous immunity is to dengue 1, and the exposure to this different serotype may result in more severe disease. Right now, about half of the cases are hospitalized, and is what we have seen in previous epidemics, but we will see what happens.
Thanks for the nice talk. So the increase of the dengue cases is related to travel increase and also the climate change. So can you talk a bit about like what are the environmental factors that affects or impacts the increase of dengue, and is it particularly for any serotypes that's more circulating because of the climate change?
Yes, we were talking with Anna before that. With these questions from the media, they're always asking, is this climate change? And I think that rising temperatures are definitely playing a role. They make the mosquito be there in some places year round. And winter has sort of impact in areas like Florida, for example. The mosquito population all dies, so the virus cannot continue transmission. So having the ability for the mosquito to persist year round, that with rising temperatures, that seems to be more of the norm, that definitely impacts transmission. The other thing is that higher temperatures also facilitate reproduction of the virus inside the mosquito, increases viral load, and increases the probability of transmission. Extreme weather events like hurricanes and floods also contribute to an increase in breeding sites. Usually after a hurricane, the population of mosquitoes comes down because of the strong winds, but after a few weeks, they go up. So those extreme weather events also play a role. However, that's just part of the puzzle. As I was saying, travel, increased urbanization play a role, and in areas with really good sort of housing conditions, air conditioning, screens, houses that are built separate apart, good services. Even if you have the mosquito, the right temperature, you may not have explosive outbreaks.
Thank you. We're going to move on to our second speaker, who is Dr. Anna Durbin, who I hope many of you know. Dr. Durbin is the director of our Center for Immunization Research, and she has been working on flaviviruses, and most particularly dengue, although also a little bit on West Nile and certainly on Zika, really since she came to Hopkins. She has developed a human challenge model, a controlled human infection model for dengue, which has been really useful both in the development of the NIH Merck Butantan vaccine, where all of the trials, the early stage phase 1 and 2 trials, were spearheaded by our Center for Immunization Research, but also for the development of a dengue antiviral that may come to licensure and may be available for treatment. She's served on a number of advisory committees for the FDA, for WHO. Dr. Durbin, please.
Thanks so much, Ruth. And I have to say it was really, what did I do? I see the slides, but I don't see them. I see them here, but I see Gabriella's slides, but not my slides.
Where's your presentation?
It's probably behind. See if it's behind or if that's, yep, that's me.
This is you?
Yep. And this advances.
Yes.
Great.
Or here.
Maybe I'll just do here. It'll be safer. So I'm going to talk about dengue vaccines, and I'm sure I'm looking in the audience to see students, former students, present students who've heard me talk about dengue vaccines. But if you've been in my class, you know it's complicated. And we're going to go through some of the complications that we have. Gabriella did such a nice introduction, and so I'm not going to have to repeat a lot of this. You'll recognize the figure at the bottom. But what I always start with is just a reminder, a little bit of a reminder that when we talk about a dengue vaccine, we're really talking about 4 individual vaccines because there's 4 individual viruses. And for the reasons that Gabriella discussed earlier, we really do need a vaccine that is safe and effective for each of the 4 individual serotypes. And that's been really difficult to achieve. We're hoping with a vaccine that will be able to induce long-lived immunity to each of the 4 serotypes, protective immunity at least against the more severe forms of the disease. We're not asking for sterilizing immunity, which is a really high bar. But what we have learned, for the reasons Gabriella mentioned, is that partial immunity to dengue is bad and can be problematic and lead to more severe disease with future infections. I keep messing this up. I think another important point that we had with dengue vaccine development is that we don't have a good animal model. So we can give a vaccine in animals and we can look for an immune response, but it's not necessarily predictive of protection. We saw that with early vaccine candidates that were essentially subunit protein vaccines of the envelope protein that looked really immunogenic in mice, but did not provide any really significant immunity in humans because mice identify different epitopes than humans do. And so we also don't have an animal model that replicates disease. So we can't test our vaccines to see if they're going to induce down the road more severe disease. So that's been a bit problematic. And we've always thought that neutralizing antibody is really indicative or predictive of protection. But I'm going to show you some of the results in the early clinical trials that really turned our head around and said, we can't necessarily rely on neutralizing antibody. So I'm going to talk a little bit about early dengue vaccine work. And I'm at the Center for Immunization Research. I know other people are. I won't put you on the spot, but does anybody know who this is? He's a very famous vaccinologist. This is Albert Sabin. And before he worked on polio, he actually worked on dengue, and he identified dengue serotypes 1 and 2, which he called dengue serotype 1 was he isolated in Hawaii, and serotype 2 was New Guinea C. He had New Guinea A, B, C, and D, and it turned out that New Guinea C was dengue 2. So he, during World War II, as the Army often does, is looking for vaccines to protect soldiers. Mouse passaged these viruses, particularly the Hawaiian strain, which is dengue 1, in mouse brain. So he would inject mice in the brain and then recover the virus, passage it again, recover the virus, passage it again, until he finally found one that was attenuated. And he noted that after 7 to 10 passages, the illness was modified. It wasn't asymptomatic illness. I'll tell you that when you look at his early papers. There still was fever, there still was some rash, but certainly modified. It did not move forward with Albert Sabin after World War II, but other investigators did take it forward. And Japanese scientists were doing the same work. And there was a large outbreak in Osaka in 1943, and they again passaged the virus in mouse brain. They didn't know what strain of virus it was at the time. But what's interesting and why I'm presenting this is a paper that really shows you can have very long-lived neutralizing antibody to dengue years and years after inoculation or infection. So 40 years later, they brought people back who were in these original experiments, and they measured neutralizing antibody, and they showed that the virus they were infected with was the mouse-modified dengue 1 virus. And 40 years later, they still had what we would consider to be very good titers of virus. And this, again, is something where we think live attenuated vaccines can induce long-lived, we hope, protective immunity. So the current vaccines that I'm going to talk about today are all live attenuated vaccines. They're called tetravalent, meaning they have 4 components. At the end of my talk, you can decide whether they really are tetravalent in action and not just in name, but we'll talk about that in a little bit. Some of the reasons we like live attenuated vaccines is because the vaccine presents your body with a modified pathogen that doesn't make you sick, but your immune system sees that pathogen or sees the vaccine as it would the pathogen. So you get a very good Th1, Th2 immune response, you get good CD8 T cells response, you get good humoral immunity. They're generally less expensive to produce, but they can be very difficult to develop, which we'll talk about in a minute. You get long-lived protection. Think about measles vaccine, think about yellow fever vaccine, but they are contraindicated in certain populations, so they're certainly not for everybody. But I think the most important point I want you all to remember is that for a live attenuated vaccine to work, that vaccine has to infect and it has to replicate because your body is the vaccine manufacturing plant for that vaccine. We give such a small amount as an inoculation, we're betting on that virus to replicate within the host and be presented in the immune context that the pathogen would so that you get a very good, broad, long-lived response. So these are the 3 vaccines that I'm going to talk to you about. Denvaxia, most of you may have heard about Denvaxia. I think it really is, I always say this, we learned more about dengue, I think, from the Denvaxia vaccine study than we ever learned about all of the studies we've done in mouse and other studies. We learned a tremendous amount. And there's certain things that I want to point out with this. First, you'll notice the color coding. Yellow is for yellow fever, 17D vaccine, because Denvaxia is a chimeric. It's part dengue, part yellow fever 17D. All of the non-structural proteins are yellow fever 17D. We think the envelope protein of dengue is the important antigen, the protective antigen, and that's indicated by the individual colors of dengue 1, 2, 3, or 4, along with the preem. So we have the preem and E proteins replacing those of yellow fever and the yellow fever 17D background. You'll notice that this vaccine requires 3 doses to be given over the course of a year. 3 doses given subcutaneously. And that's very unusual for a live attenuated vaccine. To be given systemically and require 3 doses is unusual. It's authorized now by FDA and ACIP for people 6 to 16-year-old who are living in highly endemic areas of the US, which currently is Puerto Rico and the territories that Gabriela mentioned. WHO recommends it for 9 to 45 or older. It also requires previous documented infection with dengue, either by a blood test or a clinical evaluation with documented infection in the past. So you have to have had dengue to receive this vaccine, again, which is unusual. The other 2 vaccines, one is the Takeda TAC-003. It is also a chimeric for 3 of the 4 serotypes, but it's on a dengue 2 background. It requires 2 doses to be given over 3 months, and it's currently recommended by WHO for people 6 years of age and older. And then TB-003 is the vaccine that I've been working on for 25 years now. It was developed by the US National Institutes of Health, but the technology's been licensed to Butantan, Merck, Serum Institute of India, Panacea, and other companies, and we'll talk about this in a bit. So I think what the points I want to mention here is this does not contain any non-structural proteins of dengue. They are all non-structural proteins of yellow fever 17D. And work that's been done by Alex Sete and Daniela Weisskopf have shown that more than 85% of the CD8 T cell epitopes are located in the non-structural proteins. So you get a very nice CD8 T cell response to yellow fever, but not to dengue. Phase 2b efficacy study we're going to talk about first, and that was conducted in Thailand in children 4 to 16 years of age, and this is what changed the dengue world. So we see, and what I'm highlighting here are the neutralizing antibody titers to each of the 4 serotypes 1 month after the last dose of vaccine. So 1 month after the 3rd dose, and what we see is the percentage of people who are seropositive, 95% to 100% to each of the 4 serotypes, with what dengue people would consider to be very good neutralizing antibody titers. 2 phase 3 studies started while this phase 2b was ongoing. One was in Asia, in 5 countries in Asia, in children who were 4 to 16 years of age, and one in 5 countries in Latin America, in children who were 9 to 16 years of age. Before those phase 3 studies ended, we had phase 2 results, and these are the phase 2 results on top here. And what you can see is, in this phase 2b study, the vaccine did not produce significant efficacy. They didn't look, it was not powered to look at hospitalization or severe dengue. It was a smaller phase 2b study, but this shocked everyone. Everybody predicted this vaccine would work based on the neutralizing antibody titers that it induced. The company continued the phase 3 study, and we got better efficacy, and this is against virologically confirmed dengue of any severity. And we see better efficacy, significant efficacy here, about the same that we see with flu vaccine. But what I really want to point out is the efficacy against hospitalization and the efficacy against severe disease. And you saw the pictures that Gabriela presented about hospitals being overwhelmed in dengue epidemics. This would be a very significant public health impact. If you can prevent 80% of hospitalizations for dengue, you can really have a very important public health impact. So, from these results, people thought that the vaccine would in fact have a very good public health impact. But when they did a little bit deeper dive into the analysis, what they saw was that there was a significant difference between efficacy if you've already had dengue compared with if you've never had dengue. So the vaccine did not induce significant protective efficacy against virologically confirmed dengue if you've never had dengue. It had very good efficacy against virologically confirmed dengue if you've already had dengue. And just sort of think about it, if you've already had one dengue infection, Gabriela showed you, you have much less severe disease with your 3rd or your 4th dengue infection. So if you've already had dengue and vaccination is your 2nd infection, you've really broadened your immune response to have greater protection against more severe dengue should you subsequently been infected. But if you've never had dengue, the vaccine is acting as your 1st infection. So if you don't have good balanced protection, you may be set up for more severe disease if you're subsequently infected with dengue. And that's exactly what we saw in the youngest kids who were enrolled in this study. This was in the 5 countries in Asia where they enrolled kids very young. You can see in kids who were 2 to 5 years of age at the time of vaccination, in year 3 of the study, which is 2 years after they've completed their vaccination series, they had a 7.45 relative risk of hospitalization due to dengue if they were vaccinated 3 years previously. So the vaccine gave them 7.45 relative risk of hospitalization for dengue if they were subsequently infected with dengue. You can see that as you got older, that risk went down. So the big question was, is this an age phenomenon? Is this a serostatus phenomenon? Now, they didn't draw pre-vaccination blood samples in everyone, and only about 10% of volunteers got a pre-vaccination blood sample. So they couldn't do this analysis on pre-vaccination blood samples, but they were able to develop an assay later on to try to answer this risk. And this was one of the requirements that SAGE said. So what we saw was variable efficacy based on serostatus, based on age, based on serotype. Very good efficacy against hospitalized dengue and severe disease, but there was an increased risk of severe disease in the youngest of kids. And what we found out later through an assay that was developed was that that risk was determined to be serostatus at vaccination. So now we know on an individual level, if you've never had dengue, you're at elevated risk for more severe disease. But on a population level, we can have a very good impact against hospitalization and severe disease. So this vaccine was in fact licensed, and in April of 2016, it was launched in the Philippines. And it was launched following guidance by SAGE that said this should only be used in highly endemic areas in kids 9 or older. So in April of 2016, it was launched. In November of 2017, Sanofi relabeled the vaccine to be used only for those children who've already had dengue. And then in February, it became a national scandal. And it was really devastating across the Philippines, not just for dengue vaccination, but what you can see here is that all vaccinations, the confidence in vaccines fell dramatically in the Philippines because of this episode. And it wasn't that really there was a lot of new information out there at the time. It was how this information was perceived and how the relabeling of the vaccine for only those who already had dengue was perceived by the public. But it was devastating. The Philippines is still recovering, trying to recover vaccination rates because of this episode. So what did we learn? We learned that neutralizing antibody is not predictive of vaccine success, but that we need to test for serostatus before vaccination. And now the recommendations from regulatory authorities is that everybody needs to have a pre-vaccination blood draw. We also know that, or we think that CD8 T cells may be important in the mitigation of disease of dengue, and that we now think that it's important to have CD8 epitopes in the vaccine. And we also learned, very importantly, as we've all learned through COVID and many other vaccine incidents, is that messaging and transparency are very, very important. We have to gain and we have to maintain the public trust if we're going to use these vaccines. So I'm going to move on now to TAC-003. This was a phase 3 clinical trial that was conducted in numerous countries in Asia and Latin America. They enrolled more than 20,000 people. 27.6% were dengue naive at the time of enrollment. And that's important when we look at the different results to these trials, because these vaccines behave differently for the most part in people who've already had dengue and those who've not already had dengue. So this vaccine has excellent efficacy in year 1 against both virologically confirmed dengue of any severity as well as hospitalized dengue. And it performed well in year 1 in both those who were seropositive and seronegative. What we see as we go down, this is year 1, this is year 2, year 3, and then cumulative data for the first 3 years of the study. And what we see here in seronaives is that efficacy against virologically confirmed dengue declines over time in seronegatives, and efficacy against hospitalized dengue declined over time in seronaives and was not significant in year 3 of the study. Now, this was driven by a particular serotype, dengue serotype 3, and that's something that we're going to talk a little bit about. But I think it's important to remember that these numbers, these different serotypes, you can have efficacy against different serotypes, and that can affect your overall efficacy, and it's important to note. So what you see here is even though in year 1, you had 82% efficacy against virologically confirmed dengue in seropositives and 74.9% in seronaives, what you see is that is varied by serotype. So this high efficacy is really driven by dengue 2 over each of the 3 years. You can see that very good efficacy against dengue 2 is maintained over the 3 years. And again, this is the only full-length dengue vaccine in the vaccine. The other components are chimerics. We do not see significant efficacy against dengue 3 or dengue 4 in people who have not had dengue before. And we're losing efficacy to dengue 1 over time in seronaives. So we're seeing a difference in the protective efficacy of the vaccine in seronaives to 3 of the 4 dengue serotypes. When we look at year 3, and again, this was driven by dengue 3. This was driven by dengue 3 and particularly hospitalized cases of dengue 3 in one country. But what you can see is no overall efficacy against hospitalized dengue 3 in seropositives or seronegatives in year 3 of the study. And in seronegatives, you can see the confidence intervals are quite large. Now, part of that is there are a limited number of cases of dengue 3, but it is a signal that people are currently watching, and SAGE has asked for additional information to try to get a better handle on what this may be due to. So we have, again, excellent efficacy against hospitalized virologically confirmed dengue in dengue-naive subjects for 2 years post-vaccination. But again, we're seeing varying efficacy by serotype and serostatus, and declining efficacy over time, particularly in those who seronaive. There was a signal, although it was not statistically significant, for dengue 3. There were excess cases of dengue 3 in year 3 of the study, hospitalized dengue 3, again, driven really by hospitalization in one country. But again, we're seeing an overall population benefit. You could have a huge impact with the protection against hospitalization and severe disease, with a potential individual risk, and how do we balance that? This was, SAGE met less than a year ago, September of 2023, to discuss this. And what they decided was that they're going to recommend introduction to children aged 6 to 16, even though the study was down to age 4, in settings with high seroprevalence of dengue, which was defined as a seroprevalence of 60% or greater by age 9. This is a 2-dose schedule, 3 months apart. It did not recommend, SAGE did not recommend vaccination of children 4 to 5 years of age because of the low performance of the vaccine in this age group in both seropositive and seronaives. They recommended post-licensure studies to provide more precise estimates of the effectiveness and risk profile against dengue 3 and dengue 4 in seronegative persons. And they said travelers with previous dengue vaccine should be vaccinated. Travelers without previous dengue vaccine should be told that the vaccine may not work against dengue 3 and 4. There is no testing requirement prior to vaccination for this vaccine. The company withdrew its BLA with the FDA, so this vaccine, as Gabriella mentioned, will not be licensed in the US in the near future. We'll see if the company decides to resubmit their BLA to the FDA, but we won't have this vaccine available in the US for several years to come. I'm going to move now on to the very long Butantan, NIH, Merck, Serum Institute vaccine. I'm just going to call it TB003 because it's shorter. But it is a single-dose vaccine. The phase 3 trial was evaluated in people aged 2 to 59. The trial was only done in Brazil. It was sponsored by the Institute Butantan. And you'll see that that is a bit of a problem when we look at the different serotypes that were circulating in Brazil at the time. I do want to comment that we have 3 full-length dengue viruses here. We have 1 chimeric virus, the dengue 2, 4 chimera, where we have the preeminence of dengue 2 on the dengue 4 background. So this, I want to say this is, I think, one of the best examples of your tax dollars at work, and I hope you'll appreciate that after you see the results of this trial. But we were able to do, I think, about 25 clinical trials of different monovalent and tetravalent components of this vaccine because the NIH sponsored those trials. Neither Sanofi with Denvaxia nor Takeda with TAC003 evaluated the individual components of their vaccine before putting them into a tetravalent admixture. So we have no idea if each of the individual components of those vaccines actually infects and replicates. We did more than 25 trials to identify the best monovalent components and then put those monovalent components into 5 different tetravalent admixtures to choose the best one that can move forward. This was all funded by your tax dollars at work. And I think it's going to be really, really beneficial. So this is the Institute Butantan phase 3 clinical trial. I just want to point out here, 47%, almost half of the individuals enrolled in this trial were seronaive to dengue prior to vaccination. And that's going to be able to give you a really good evaluation of how well this vaccine is going to perform in dengue-naive people. These are the preliminary interim analysis, the interim analysis results that were just published in the New England Journal of Medicine just a couple months ago. What we're looking at, the overall efficacy was almost 80%. It was almost 90% against dengue 1 and 70% against dengue 2. I will say dengue 2 is the weaker component, and we know that just from different studies that we did. It's a chimeric, and it has a higher human infectious dose 50 than the other components. When we look at the differences between seropositives and seronaives, we still see very good overall efficacy in seronaives to both dengue 1 and to dengue 2. Again, a bit lower, but still almost 60% efficacy through 2 years post-vaccination. There was not a 1-year interim analysis event in this study. They did not do the analysis until everybody had completed 2 years of the study. This is what I think is really impressive, and I'll show you another slide in just a bit that compares the different vaccines. But in the very youngest kids, 2 to 6 years of age, through 2 years, we have 80% efficacy. These kids, 82% of these kids were seronaive at the time of vaccination, and 73% efficacy in seronaive. So very good efficacy in the very youngest of kids through 2 years post-vaccination. There were not any cases of severe dengue in the first 2 years. They did present through the cutoff, which was an average of 3.7 years post-vaccination in different conferences. And what they saw was that there was 88.2% efficacy against severe dengue and dengue with warning signs through 3.7 years post-vaccination. So quite a long time post-vaccination, very good efficacy against severe dengue and dengue with warning signs. When we look at the 3 different studies, again, with Sanofi, this was really at 1-year follow-up. They only follow-up, which is why there's an asterisk here in the different age groups, and just showing different dosing, 3 injections, 2 injections, 1, and then looking at efficacy by age. And again, in the youngest age group, this is really where I think this vaccine stands apart, is its efficacy in the very young, 80% of whom were seronaive to dengue at the time of vaccination. And then when you look at the 3 different vaccines by the serotypes that we have, again, there were not any dengue 3 or dengue 4 cases in the Butantan trial for the time that this was presented. Dengue 3 started circulating a little bit last year. Now, all 5 years of follow-up is going to be completed in June of 2024, so it's unlikely they'll have enough dengue 3 cases to really show efficacy. We're hopeful, but it's unlikely. But what you can see is that very good efficacy, again, in seropositives as well as seronaives, very good efficacy against dengue 1 and dengue 2. I think I'm going to say this at the end, and then if we look through greater than 3 years, so again, this is an average of 3.7 years post-vaccination, very comparable overall efficacy, very good efficacy in seropositives and seronaives, very good efficacy against dengue 1 and dengue 2. Again, there's no cases of dengue 3 or 4. I will tell you my personal opinion is that the efficacy against dengue 3 and dengue 4 will be higher than that of dengue 1 or dengue 2, and that's based on the data here. I had said earlier that for a live attenuated vaccine to really induce a good homotypic immune response, you need to be able to infect and replicate. This is the recovery of vaccine viremia by each component of the vaccine post-vaccination. This is by PCR, and then in parentheses is by culture. But what you can see is we have imbalance in the recovery of the individual components with Denvaxia and with TAC-003. Dengue 4 dominated in Denvaxia and had the highest efficacy to that serotype. So the greatest efficacy for Denvaxia was against dengue 4. The greatest efficacy against TAC-003 was against dengue 2. Now, this viremia data is from a phase 2 clinical trial. They reformulated but didn't repeat the viremia studies. So we don't know really in the phase 3 formulation if this is different, but you can see that dengue 2 is really, really dominant. And then when we look at TB-003, this is a study that we did here in the US. We had very balanced viremia by PCR across, and you can see greater recovery of dengue 3 and 4 than even dengue 1 and 2. When we look by culture, again, it shows us that the dengue 2 component is weaker than the other 3, but we recover all 4 with higher recovery of dengue 3 and 4, which leads me to believe, but until we see the efficacy data, we won't know that efficacy against 3 and 4 is going to be even greater than 1 or 2. So in summary, we have a single dose vaccine, which is always preferable in terms of deployment and compliance because it's one and done, had very good efficacy against virologically confirmed dengue 1 and 2 through almost 4 years post-vaccination with excellent efficacy against severe dengue and dengue with warning signs. A single dose provided very good efficacy in all age groups, regardless of serostatus. And again, I think we're going to have good efficacy against dengue 3 and 4, but until we see those data, we won't know. Butantan is planning to submit to Anvisa in late 2024. There's rumors going around, Gabrielle and I were talking about it earlier, that the Brazilian government may deploy TB003 earlier to meet the severe epidemic that's ongoing in Brazil now. Anvisa has modified its to have a rolling submission. It used to be you couldn't have a rolling submission, but because of the urgency to get dengue vaccines licensed and used in Brazil, they went to rolling submission, and we'll see how soon this vaccine can be ready. The problem is you have to have supply, and I think that is an ongoing question. I am not convinced that they will have supply that will come close to meeting demand for the next couple of years. These are the licensing partners that NIH had. David, they did license to a company, Incepta, in Bangladesh, and that company is very interested in producing vaccine for Bangladesh, which is also having a really terrible, terrible outbreak. So these are the lessons learned. I think chimerization is highly attenuating and maybe over-attenuating for some viruses. Again, for live vaccines to induce immunity, we have to infect and replicate, and we need balanced homotypic immunity. And that's been very difficult to measure because we have cross-reactive antibodies. And Gabriela hosted a meeting at CDC Puerto Rico just a few weeks ago, trying to say, can we come up with an assay that can distinguish between protection and risk with these dengue vaccines? And that's something I think that we really have to work on. And there were a lot of very smart people in that room who are working on that now. I do think that T-cell immunity is important for mitigating disease, but of course it's very difficult to have a standardized assay to measure that for supportive approval. So other considerations, and I think we're going to talk about some of this in our panel discussion, is how are we going to ever have enough vaccine to meet the global demand? The global demand is only going to grow, and I think we only can supply a minute fraction of that right now. There are additional manufacturers that are going to come online in the next 3 to 5 years, but that's still 3 to 5 years away. That's not tomorrow. And then how do we integrate these vaccines into a vaccination schedule that can facilitate uptake? For some of these vaccines that require 3 doses or 2 doses and ages in adolescence or higher, that may be difficult. And then really importantly, how do we monitor or how do we follow for long-term safety signals? And I'll end here. Thank you.
Gabriela, please. Why did you use the chimeric virus for the dengue 2 component? That's one question. The second quick question is, is 50% efficacy for dengue 2 among seronegatives enough? And what about that 50% that doesn't have protection?
So,
Do you have enhancement?
So the first question is, why did we use a chimeric? And that's a great question. Early days, we tried many, many different components. And we, as I said, we looked at them individually. And the NIH's original strategy was to have a chimeric dengue 1, 2, 3 on dengue 4. But the chimeric dengue 1 didn't work. The chimeric dengue 2 in non-human primates looked attenuated but still induced an immune response. So it went forward into human studies. And then in human studies as a monovalent, it's still, although it infected, we had about 80% seroconversion with that. So we moved forward with that. We had a 3-4 chimeric, which was over-attenuated and was tossed. And that's what prompted different dengue 3 candidates, including the dengue 3 delta 30, which was under-attenuated, and the dengue 3 delta 30, 31. And I said to Steve, I think, if we had that 10 years ago, we may do a dengue 2 delta 30, 31, but it was too far advanced. And we do think the second question was, is 50% efficacy in seronaives at 3.7 years, is that enough? And I think against virologically confirmed dengue, it is if you have, again, homotypic immunity. So I think what was more compelling or encouraging was at 3.7 years seeing almost 90% efficacy against dengue with warning signs and severe dengue, meaning that even if you're not preventing infection, you're preventing the more severe complications. But that's something that's going to have to be followed. And it'll be interesting to see if there are second generation vaccines that come down the pike and they replace that. There's other concerns about even different strains and strain variation and how will that affect efficacy and things like that. And I think we're going to continue to learn from dengue for many, many, many years after these vaccines get deployed. Maria.
Hi. So interesting. Probably not my field.
But in my field, we have indirect effects. And so I'm really curious, if you reduce infection, can you reduce transmission within the household or otherwise?
That is a great question, and people are looking at. And one of the questions is, do we want to completely reduce transmission? Because we may get natural boosting, which could provide additional protection for many, many years. I think where there is greater indirect protection, and it's something that I alluded to earlier, is, if you can just prevent hospitalization and severe disease, I think you're making a great contribution. And there was a lot of debate about both Denvaxia and still debate about TAC 003 with that. I mean, Denvaxia could have had, I think, a huge impact in terms of hospitalization and severe disease in the most affected countries. But you have that ethical dilemma of when you've identified an individual risk, are you then compelled to avoid that risk? And the answer was yes. So now we have to test everybody before they get vaccinated. And Gabriela will tell you that Denvaxia uptake in Puerto Rico has been terrible for many different reasons, right? It's just been very, very low. And there's some, and you can confirm this, that Sanofi may stop making if they haven't stopped producing Denvaxia because uptake is so low. Yes.
So I have a question for you. So thinking about going back to thinking about the fact that dengue 2 is chimeric in this vaccine, you spoke a lot about how you think that the T cell response is important for immunity and that the T cell response is predominantly to the non-structural proteins of dengue. So do you foresee there being any issues down the pike with homotypic immunity dengue serotype 2 because it's on a dengue 4 background?
That's a great question. I have so many slides I could show you. But so we did a study where we looked at the CD8 T cell responses to individual monovalent components and the CD8 T cell response when you give it as a tetravalent. What's really interesting is when you give a monovalent, most of the CD8 T cell response is directed against serotype-specific CD8 epitopes, so those individual. When you give the tetravalent, that response moves to conserved epitopes across each of the 4 serotypes. So the tetravalent, about 80% of the CD8 epitopes that were recognized by after tetravalent vaccination was conserved across all 4 serotypes. So I don't think it'll be a problem because we have the 3 other serotypes. We did other experiments, but I'll tell you later.
So that's great. And I think what we're going to do is move to our panel discussion. So I'm going to ask Anna and Gabriella to come up along with Holly and Morgan. And Anna, you sit there, second seat. I mean, it doesn't really matter. People want me to read something over my head. Oh, yeah, that's fine. That's really good. I guess I have to go to the podium. And what I'm going to do is I'm going to first, you now know Gabriella and Anna, but I'm going to ask our other 2 panelists to introduce themselves, if they would. Morgan and Ruth Holly.
Yeah. Hi everyone.
And
My
do
name
you
is.
have microphones?
I think
Right
it's
there.
I think it's
Okay.
working. There's one right
Cool.
here.
Great.
Hi everyone. My name is Rupali LeMay. I'm an associate professor in the Department of International Health.
Hi everyone, my name is Morgan Park. I'm a second year MSPH student in the GDEC program. I intern with PAHO on the vaccine safety team, specifically looking at the dengue vaccine.
Great. So what I think we're going to do here is I'm going to warm the panelists up with a few questions, and then I think we're going to turn it back to the audience to ask some questions. And I think I'm going to actually start with both Gabriella and Anna. So for countries that are introducing Qdenga now, which is really what's available, I mean, the TV003 is maybe going to become available in Brazil, but probably not in other countries. So for right this moment, with this huge explosion of dengue, the choice is Qdenga. So what are the implications for these countries? I mean, how do they introduce? What are the public health measures that need to be put in place? What are the considerations for regulatory authorities for NITAGs? How do they think about this?
That was like 10 questions in one.
You can. So what I would say, Gabriella, is you can choose one. You can choose your favorite. Yes, you can choose your favorite question.
That sounds great. And I think now several regulatory agencies have provided approval for Qdenga, and several individual countries as well. I think Brazil is sort of leading and providing the thought process about how to implement it. They're prioritizing the largest cities with the highest dengue historical burden, which is great. They are strengthening their surveillance system. They have named reporting. That is not the case everywhere. In many countries, they report in aggregate. And in terms of monitoring safety, is not the regular safety monitoring that you have with other vaccines that you monitor the next 24 to 48 hours after administration. This is knowing what will happen years later. So it's very challenging. And one way of doing it that we were discussing earlier is sort of thinking about how to be able to combine the case surveillance databases with the immunization registries and try to find out if people who are hospitalized later have been vaccinated. So very challenging. Many times parents cannot recall if their child received the dengue vaccine or not. And then thinking about additional studies, like potentially test negative designs. Mm-hmm. The main challenge is knowing baseline sero status. And from our experience in Puerto Rico, they were implementing the Vaxia. We have to do a lab test before administration, since only seropositives can receive the vaccine. And this requires for pediatricians to order the IgG test. Based on results, they refer the child to a vaccination center. So there are so many places where the children can be lost, right? Going to the pediatrician first, the pediatrician thinking about ordering the test, getting the results, then sending them for vaccine. Like, I cannot think about a more complicated way to administer a vaccine. So you can sympathize with me and the challenges we're having in Puerto Rico. So knowing baseline sero status for public health implementation of a vaccine through an immunization program, I think it's almost impossible. So many challenges in terms of the long-term monitoring.
Yeah. I forget which part I have to answer now. No, I'll say the other thing, there's a couple things. One, also, if you have the case, so there's very limited supply of Qdenga. I think that has to, is a real problem. So you really have to prioritize how you're going to use those doses. And I think, that's going to be difficult. As I said, demand's going to far exceed whatever we can provide. And so I think you're going to have really good messaging and really good strategies around how you're going to deploy that. And also the other issue is anytime you have multiple doses, and how do you ensure that vaccination series is completed? I think it is interesting. We talked a lot about this for COVID. This is the first time Gabriella's heard me say this because I just thought of it today, but thinking about how you stretch doses, so this is a 2-dose series 3 months apart. They're coming to the end of their dengue season now. So it's unlikely that any dengue vaccine that you administer in Brazil now is really going to have an effect. Can they have enough production so you can vaccinate more people with the first dose now, with the idea that you'll have more production and can then get them their second dose 3 months from now? Or do you say we're reserving these doses for the people who've gotten one dose so we ensure that there's enough for a second dose. And I don't know what the answer is. I think that, I don't know what Brazil's doing, but when you have a limited supply and a high demand, it can be really, really difficult.
So Anna, I'm going to follow up with a question for you, which is, you and I stand up in front of classes and we say, oh, live attenuated vaccines are really easy to manufacture and you can make a lot of them. And, I can just tell you, as you know, and as we've discussed, right now dengue is on the Gavi shortlist for and with good reason, because of all the global outbreaks. So, can you tell us more about why it's so hard to make global supply and what would be needed to really, it's great that these other, I think this is the wave of the future and kudos to the NIH for pursuing licenses with all of these global partners. But what can be done now to enhance global production?
Give Butantan lyophilizers. That's number one. But and I'm only half joking. That is the bottleneck right now, is lyophilization and capacity. We said, can Serum Institute, who could lyophilize Rhode Island, can they help? Things like that. But I think really it's going back to COVID. What do we all do, right? What do companies do? They don't scale up and manufacture until they're well into their phase 3, and perhaps even until, they have to do the lot-to-lot consistency study. So these companies don't scale up manufacture until they're almost at licensure, and then there's not the supply to meet the demand. And I will tell you, I've been going down to Butantan for 15 years or more, and every time Steve and I would go down there, it's, you have to make sure if you get approved, you have enough vaccine. If you get approved, you have to have enough vaccine. But companies don't want to invest in that infrastructure until they're assured they're going to have a product. And I think that's part of the problem. I'm encouraged, and this won't solve the issue today or tomorrow or probably for 2 years, but I do like the idea of licensing out the technology to different in-country manufacturers so that you have multiple manufacturers that can produce a vaccine in their country and for other countries. That won't take effect probably for 5 years until, as I said, Serum comes online, Panacea comes online, Merck comes online. But you need multiple manufacturers for almost any vaccine if you're going to meet demand in an epidemic or in a large outbreak.
Thank you. So Morgan, I was wondering if, first of all, I think it would be really great if you could briefly tell the audience about some of the work that you have done with PAHO around dengue vaccine safety assessment and some of your thoughts about why in the vaccine safety world, assessing dengue vaccine safety is so much harder than typical vaccine safety assessments.
Yeah, so my largest task during my internship at PAHO was just conducting a safety analysis of the vaccine. So any pharmaceutical company will conduct a plethora of clinical trials. For Qdenga, it's been 19 so far, 14 of which have publications available. And it's a really hard and complicated task to go through and comb through all of these efficacy data, all the safety data to figure out what is kind of the profile of this vaccine. In October of this past fall, SAGE recommended the vaccine, and a lot of countries will just take this recommendation and run with it. But because they don't have the capacity or they don't have the personnel or the time or all of the above to really go through all of the safety data themselves and apply it to their country-specific context. The benefit of being an intern and having time to do things that other people don't have time to do was I was able to go through and essentially pull every single piece of safety data from these clinical trials and put it into a summary findings table. This summary findings table will then be included in PAHO's guideline for conducting post-licensure vaccine safety surveillance. And the objective is really just to provide countries with all of the data in a really digestible way so they can make decisions for themselves. without having to do all the work that I got to do. I say got to instead of have to because it was very valuable. In terms of why dengue vaccine safety surveillance is so different than other vaccines is because what we're really looking for and what we saw with Denvaxia was a potential increased risk of hospitalization in seronegative vaccine recipients. And so we really need to make sure that we are not having breakthrough cases lead to a higher risk of disease. So in terms of vaccine safety surveillance, instead of looking for adverse events following immunization, anywhere from a minute to weeks after immunization, and we're talking about years in the future. And the adverse events that of interest is really the disease itself. And so when conducting vaccine safety surveillance, Gabriela's mentioned this earlier, you really have to have interoperability between disease surveillance systems and vaccine safety surveillance systems. And these two systems frequently don't communicate with each other, or one is significantly more developed than the other in terms of the Americas. And because we're used to dengue, our disease surveillance is so much more robust than our vaccine safety surveillance. And it seems really simple, the idea of these two systems communicating, but they often use different software, different information systems, and they're also managed by different agencies within one country. So even within a country, sharing this data is hard. And then when you look internationally and sharing data with PAHO or sharing data with neighboring countries, that's even harder. So dengue is very unique in a sense that what countries need to do is really conduct vaccine surveillance, vaccine safety surveillance, and disease surveillance at the same time, and also have really robust vaccine registry systems to know that if someone comes in with dengue 3 years after vaccination, we know what type of vaccine they got, so we really know the true efficacy of vaccines in the future.
Thank you. Okay, so Rupali, I'm sure my guess is that I can ask you questions, but my guess is that you've been spotting issues as you've listened to these other presentations. Communication challenges. I think there's probably a laundry list of what these might be. And I wonder if you might just want to pick a couple that particularly resonate with you and talk about how you would communicate to a population, how you think about population versus individual risk, this stuff that Anna brought up. But please take it away.
No, thanks, Ruth. I think the biggest challenge, and Anna brought it up as well, I think is really this idea that demand is going to be way ahead of supply. So to me, I think we learned that during COVID with regards to the allocation frameworks that were developed during that. And then even in addition to that, and subsequently thinking about the malaria vaccine products, that has been a huge challenge to think about with regards to how are we prioritizing, who are we prioritizing? And then in addition to that, many of us are working, I think, to prepare for TB vaccines. And that's going to be even a larger issue with regards to the amount of morbidity and mortality that tuberculosis really does affect in a number of countries and a number of individuals all around the globe. And so I think the first is going to be, thinking about the communication challenges that we learned during the COVAX allocation framework, how we are going to apply that and adapt that to dengue, I think is going to be critical. And we should think about, again, an outbreak sort of lens and perspective. I think that's the first one. The second one, I was sitting next to Morgan during the talks, and I was saying, because she knows everything about dengue and I know very little about dengue, so I was asking her questions. I mean, the piece about the partial immunity is going to be really hard to communicate. And I think thinking about, yes, we want you to get the vaccine, but we don't want you to get the vaccine if you necessarily haven't been infected before, right? If that's really what ends up happening with regards to the recommendation, I think is going to be a key challenge. And that, I think, underlying all of that is really the decline in trust towards vaccines that we've seen globally. We saw that it was already declining before COVID. COVID really, I think, did a doozy with regards to vaccine science and just vaccine confidence. So I think when any product is coming out, including a new product, there's going to be, I think, even more issues with how do you communicate to the moms, especially that are probably making the decisions as to whether or not their child would get a vaccine. But I think more importantly, even to the healthcare workers, because I think that we saw in new recent vaccine introductions, a lot of the hesitancy was really around healthcare workers not wanting to recommend the vaccine. And so I don't want to forget that population, right? We want to think about the beneficiaries, but we want to think about the provider population that we're going to be asking to really counsel individuals as to why they should get the vaccine or not.
Thank you. I think we'll open it up now to questions from the audience if we have. I wonder if you could go over to the microphones.
So hi, my name is Julie, and I have been following really closely the Brazilian Ministry of Health updates on vaccine rollout. And it actually seems like since February, they have started inviting children in a certain age range to come into the selected, cities or municipalities that are most affected. And uptake has been super low. Parents aren't bringing their kids in. And I see the second to last point on the slide here, which has a sort of embedded assumption that I want to question, which is how to integrate these vaccines into the vaccination schedule. Do we have to follow the traditional public health approach to try to ideally you'd have mass coverage, but A, we don't have enough vaccine, and B, not everyone wants to accept the vaccine right now. So why wouldn't we consider a more facility-based approach where you take all the children, right? In Brazil, it's mostly children hospitalized and dying, or dying because they didn't get hospitalized, but in any case, many getting hospitalized. Why not offer the vaccine to all children when they're getting discharged, right? Because is there a need to wait after you have to wait a certain period? I mean, what we do is so I mostly focus on malaria, right? And with malaria at the health facility, everyone in Latin America and Sub-Saharan Africa and so on the providers are encouraged to counsel people who come in and have to be treated, to explain, okay, here's a treatment, take it for 3 days or 14 days or whatever the regimen. But here, by the way, here's how to prevent malaria for next time, right? And so there's always this counseling. So why not set up a follow-up appointment, focusing on that facility-based kind of approach, and then you already know the sero status, right? So I would think that might be a more effective way to use what's currently available, rather than trying to do more outreach. It just doesn't seem like the traditional approach is going to really pan out right now.
Yeah, I think a couple points. I think one, we'll take Brazil. I mean, one of the long-term goals is to have routine immunization for dengue. And how do you do that with utilization of your man hours and things like that? They have a very good public healthcare system and that sort of thing. So I think that's their ultimate goal, is to try to reach any kid. It is a little bit problematic vaccinating after you've already had dengue because of the immunity that you get. And Gabrielle, you have short-term protection against all four, which means you could essentially inactivate the vaccine and you wouldn't have a good take of vaccine. So for a live vaccine, that's not necessarily true for other types of vaccine, but for a live vaccine. I think, in an outbreak setting like we're having in Brazil, having sort of vaccination centers isn't a bad idea, if you can do that and bring the kids. I think a bigger question, and we saw this with HPV vaccine, if you want this to be routine immunization, you have to fit it into your current public health care structure so that you can utilize all of the resources that you have. So I think that's the goal for long term. Short term, there may be other solutions that may increase uptake. And I don't know, Gabrielle, if you have.
Yeah, and I do want to add a couple of things. First, the pre-vaccination testing is only recommended for Dengvaxia, the first approved dengue vaccine. The Takeda vaccine, Qdenga, does not require knowing SER status before vaccination. It's recommended across the board. I agree that you need to wait a little bit after someone has had dengue to vaccinate because of the cross-reactive antibodies that could inactivate the vaccine. And I think different strategies can be used. You want to integrate it to the immunization program. In Brazil, they're doing older kids, 13 and 14, so it could be paired with HPV vaccination, and that could really facilitate increasing coverage. And you could also provide it at immunization centers. I do not think vaccination is an effective tool to control an outbreak. This is a 2-dose vaccine. Of course, there will be some protection after the first dose, and I think your suggestion is intriguing. But I don't think that the time to try to vaccinate the entire population is in the middle of the outbreak when the healthcare system is completely overwhelmed. This has to be done before.
Yeah.
Hi, I'm Ashlyn. I work at the CIR, and although I'm doing vaccine research now, I'm a public health nurse at heart, and I think you touched a little bit on how like COVID has changed the way people think about vaccination, and changed the nature of the misinformation and disinformation we have kind of around vaccination. I think in the United States in particular, we've seen a type of disinformation I don't think most of us could have predicted when we have things like, Dr. Joseph Ladapo just publishing false information in Florida, like falsified information about COVID. And so like the trust is hard to gain. And I wonder if you could speak a little bit to like the political context of areas where dengue is endemic and how kind of trust in the government and trust in medicine might change the way in those areas that we need to think about public health messaging. And then if you have any thoughts on like how to effectively counter that kind of disinformation here in the States, I'm always open to hearing those suggestions.
Ruth, do you want me to
Please.
Okay, that's a
Please.
really easy one. So let me start, maybe I'll start sort of backwards, right? So there is a link between political ideology and vaccine attitudes, right? And I think part of that is pre-COVID, there were really 4 reasons why people didn't want to accept a vaccine. We could summarize them down. One had to do with the ingredients. One had to do with the schedule, i.e. the number of doses, the number of vaccines that are recommended. One had to do with risk perception, meaning people didn't think they would get the disease, and they didn't think even if they got the disease, it was severe enough to get a vaccine. And then the 4th really had to do with adverse events, right? This idea that there could be a link between vaccination and, autism, even though that has been refuted. COVID then happened, and we saw new drivers come related to vaccine acceptance. So I would argue that polarization is one of them that has led to more refusal of vaccines, broadly speaking. There was an analysis that was done actually by an MPH student last year for her capstone that was really interesting. She looked at communication by state, and then she looked at ideology, meaning what was the affiliation of the governor, how many representatives did they have, were they Democratic or Republican? And what they really found is that in states where there is more conservative leadership, that you're seeing people less adherent to public health measures and a larger proportion of distrust, right? So I think it's what we would hypothesize that would happen. What we've seen in some of these cases in Philippines is a great example when you think about Dengue and you think about the government and you think about the communication that happened during the government, during especially the rollout of that. I think that to me, and I know a little bit about the Brazil context, but I'll hand it over to Ana and Gabriela as well, that I think thinking about the ideology of whether or not we're talking about any type of recommendation that's done from a public health lens, whether or not it is sort of treading on liberty is what we hear a lot. Treading on my independent choice to make a choice of what I would like to do is what we're starting to see. And I think vaccines are just a casualty of that. It's not necessarily that people are anti-vaccine, they're anti the government telling them what to do, right? So I think there is, Pew Research has been tracking trust in providers in the US for the last 20, 25 years, and it has slowly been decreasing over time. COVID then took a huge hit to that. And, I think there was this analysis that perhaps this is just a US-centric phenomenon, but that's not the case. And if you start to look at COVID and COVID vaccine acceptance among countries, and again, map them politically, ideologically, you see very similar results. I think with regards to what you could do, that's a really hard one. I think the one key takeaway I would say is that I think that when you are speaking with someone that might have different attitudes than you do, is you have to come from a place of empathy. And I think during COVID, so many people had so many concerns that were dismissed. And to me, that's a missed opportunity by the healthcare system. regardless of what that concern is, I think being able to have that conversation and being able to say, I recognize that you have concerns. Let's talk about those concerns. That to me is really it. And I think it's a long game. I think people want this sort of short term. I think rebuilding that trust is going to take repeated interactions over time. And I think that is going to be really what we're going to have to do. And we're going to have to think about alternative messengers would also be my argument. Thinking about people that maybe aren't linked to the healthcare system because there is so much distrust in the healthcare system. But I don't know if Anna or Gabriella or even Morgan want to comment on the political sort of context of Brazil.
Well, I mean, certainly the former leader of Brazil was, very much anti-COVID and COVID vaccines. He may be going to jail soon, so we'll see. But I think it's what we saw in COVID too, right? During an outbreak, there are people who are panicking and who want protection. I think you can have an opportunity there, but it's as sort of things normalize, and then you sort of see a divergence. And I agree with you. It's really conversations that take time. I think to some extent Brazil has, because of the way their healthcare system is set up, they have neighborhood healthcare workers that go out and they know people, that I think there is somewhat greater trust, particularly at the community healthcare worker, not necessarily the physician, but the community healthcare worker is an opportunity. But I do think, with all of it, it is conversation after conversation after conversation. And sometimes you make a difference, sometimes you don't.
Yeah, and I would like to add, in the case of Puerto Rico, we did a lot of formative work to understand what people were thinking and who would they trust. And healthcare providers was sort of the top category from all the in-depth interviews and the focus groups that we conducted, that they wanted the healthcare providers to recommend the vaccine and to sort of explain the benefits and the risks. The challenge that we're facing is that healthcare providers have a lot to do, and this would require a lot of time from their day to explain the complexities of dengue vaccine. So you need to provide them with the information, but also have educational aids that could make their job easier. And the political support is key, because if they are evaluated on other priorities, then dengue vaccine will be at the bottom of their list. So also having local health authorities support it and make it a priority, and even the mayors from the different municipalities, makes a huge difference. So having that political support can be key.
Yeah. And I'm just gonna add one small thing. Another approach is just having a clear communication strategy, a risk strategy. In 2020, PAHO did a survey of all the vaccine safety events that are being conducted or activities that are being conducted in countries, and very few countries have an established risk communication strategy. So what countries are doing is they're very reactive in how they address vaccine safety instead of being very upfront about safety concerns that exist, which is, I think, some of the work that PAHO is doing is we're trying to make the risks that emerge through clinical trial data more digestible to larger audiences, so people are aware of things and doesn't feel like something is being covered up, which I think is a huge concern. But it's really just having, from an international health organization like PAHO, it's really trying to have a unified communication strategy so that you're not hearing different things from different people, you're not changing recommendations, which is inevitable with vaccine science. So that's always going to exist. But it's really just countries having a plan before something happens, and communicating safety concerns with international health organizations to make sure that everyone's on the same page.
Thanks.
Chrissy. Hi, I'm Chrissy. I work at the International Vaccine Access Center. And my question is mostly maybe for Gabriella, and I was interested in learning about the non-human surveillance that feeds into ARBONET, and if there are efforts to increase any of that with concerns around changes in climate change and increased dengue cases in the US.
So you're talking about mosquito surveillance. Yes. In terms of what is done, it varies from place to place. In the continental US, Florida does a lot of Aedes surveillance, but in other places they're mainly doing other mosquito species because they're worried about West Nile. In the case of Puerto Rico, we do have good infrastructure to monitor mosquito populations. There's a unit called the Puerto Rico Vector Control Unit that receives funding from CDC and has traps placed in different parts of the island to monitor adult mosquito populations. And as part of the work that CDC is doing, we're trying to establish a mosquito threshold. So the number of mosquitoes below which you don't expect transmission. And the magic number is 3 that we're still trying to validate. But in places that have 3 or more mosquitoes per trap, then we sort of target control efforts. But that can be a lot, right? In tropical areas where the mosquito populations are very robust. So additional prioritization for vector control strategies are done based on historical case count, trying to identify hotspots. So, now that we're seeing more locally acquired cases in the continental US, there is an interest from the local health departments to have more clear guidance on what they should do, how they should target areas for mosquito surveillance, and if virological surveillance in mosquitoes is helpful. And we have seen sort of mixed results. During Zika, for example, there was a lot of testing for Zika in mosquitoes, and it didn't identify additional areas where transmission may be happening. Sort of human surveillance was enough. But for dengue, we're still sort of trying to evaluate it, and we use both human surveillance and virological surveillance to guide vector control strategies.
Thanks. David, I think yours will be the last question. Thanks.
Yeah, thank you. I'm David Sack from International Health, and been working on cholera for a long time, and the use of cholera vaccine, which has only become available in the last few years. I know that cholera and dengue are obviously totally different in terms of transmission and disease, but the similarity has to do with the shortage of vaccine and in the wide, the tremendous demand. And, there is a stockpile now for cholera vaccine, which instead of the vaccine is being allocated through 2 ways. One is through the emergency use for an outbreak, and the other is through defining hotspots in countries. And I'm just wondering, you mentioned the word hotspot, and I'm wondering whether that concept is also applicable to dengue, and can you define hotspots where you really want to concentrate the use of the vaccine?
Yeah, that's a very interesting question. I think that to prioritize areas for implementation of the Takeda vaccine, they are not taking a hotspot approach since they want to introduce it as part of the immunization program, and that would mean entire cities or entire municipalities. So I have not seen the hotspot approach used to guide vaccination strategies. For vector control is very complex, and we don't have one strategy that works. The insecticide resistance is very common in all the areas that are endemic for dengue. So you need to layer strategies one on top of the other for it to work. And that could include reducing breeding sites, education campaigns, visiting homes to have clean patios, insecticiding, larviciding. So it's a lot of work that has to be concentrated in an area. So in that case, identifying areas using case surveillance that have had higher rates of dengue in the smallest possible geographic area has been used to guide vector control strategies.
Thank you. So I think this concludes this portion of Vaccine Day. I really wanted to thank the speakers, the panelists, you, the audience.